glucagon. So what part of this endocrine section constitutes this portion? Islets of Langerhans. Islets of Langerhans has alpha cells, beta cells and delta cells and various other type of cells that are visible only by the special stains. So in the pancreas we have exocrine and endocrine part. ठीक है, exocrine part में आप क्या देखेंगे इसके अंदर? Exocrine part में क्या दिखेगा आपको? Serous acini. Now serous acini are, you already aware of, these are pyramidal shaped, yes, and with the nucleus is directed towards the base. Apex has zymogen granules which is filled with these peptides which are enzymes only, digestive enzymes, and the superior part has Golgi complex and inferior part has endoplasmic reticulum. So the inferior part is more basophilic in nature and superior, that is apex part, is more eosinophilic in nature. We are talking about serous acini of pancreas and we are talking about exocrine section. Exocrine section basically constitute serous acini and the ducts. Exocrine का मतलब यही है ना, which releases secretion through ducts, and endocrine क्या होते हैं जो अपने secretion किसके through? Directly they secrete into the bloodstream. Yes, so exocrine part has ducts and serous acini, and endocrine parts have islets of Langerhans. So this is a section of pancreas. It's basically alveolar part, the acini part and duct system. So what type cells are there? Main type क्या? Acinar cells. These are basically serous in nature. What is the first example that you have read in the serous acini? Parotid gland. So the parotid and pancreas both have serous acini, और इन दोनों में क्या difference होगा slide में वो हम last में discuss करेंगे. ठीक है? There are few discriminating factors that we should know, that this slide will be of pancreas and this will be of parotid. So they are acinar cells. They have basally located nucleus and zymogen granules are present in the apex part. Then myoepithelial cells are not present here, which are there in the parotid, in the acini of the parotid gland. Here in the lumen there are centroacinar cells. These are pale colored cells which are centrally located nucleus. These are squamous type basically, और ये कहाँ present होंगे? See, these are pyramidal serous acini. This is the rounded nucleus which is present towards the base, and these are the zymogen granules. This is the luminal part of the duct. Now the point where these ducts start basically in the intraacinar part. जो acinar का lumen है वहाँ से ही अपना ये duct system start होगा, तो वहाँ जो cell line करेंगे duct को, those are centroacinar cell, and the duct system start with the squamous type of cells. Okay, now these intraacinar cells then continue into the intercalated duct. These intercalated duct combine and form intralobular duct. Intralobular duct means they are present in the within the single lobule, and then these intralobular duct drain the secretion into the interlobular duct, which is present in the connective tissue septa. Yes, come in. And finally interlobular duct will drain its secretion into the main pancreatic duct, which is known as duct of Wertsching, and accessory pancreatic duct, which is duct of Santorini. So centroacinar cells are basically what? These are the squamous type of cells which lines the lumen of the serous acini cells, and these are the starting point of the intraacinar part of intercalated ducts. So these are the pancreatic serous acini which are visible over here. These are darkly stained. Apex is more darkly stained, eosinophilic as compared to base. They have rounded nucleus towards the base, and in the luminal area there are centrally placed centroacinar cells. Next, exocrine portion should have ducts. So we have a very systematic duct system over here. First of all, to start with, intercalated ducts will take the secretions from the acinar part, and the 1 to 2 acinar parts combine and form the intralobular duct. and then intralobular duct will combine and form interlobular. So what type of cells are present which lines the epithelium of these ducts? Yes, simple squamous in the intraacinar part, that is in the centroacinar cells, and then they are low cuboidal or again squamous type in the intercalated duct, which are the smaller part. Initiating जो होती है duct जहाँ से initiate हो रही है, those are intercalated ducts. तो basically epithelium जो है वो squamous से फिर cuboidal होगी, cuboidal से columnar and then stratified. ठीक है, तो यहाँ पे कैसी होगी? Either simple squamous or simple cuboidal, and then intralobular part has simple cuboidal epithelium, and interlobular part or the duct, it has simple columnar epithelium. Main pancreatic duct is either simple columnar or stratified cuboidal epithelium. Okay, so epithelium changes with the increment of the duct system. With intercalated duct, it is simple squamous or low cuboidal, then it is again simple cuboidal in the intralobular, and interlobular we have simple columnar epithelium. So this is, see, these are the serous acini, these are the lobules. अभी तक तो समझ में आ गया हो कि हर एक जो organ होता है, it is covered by the capsule, then the connective tissue septa, it penetrates into the organ as connective tissue septa or trabeculae, which divides the parenchymal part into different lobules. So there are different lobules, and how to recognize these lobules? This is the connective tissue part or the septa which is separating the lobules of the glands. Okay, these are the different lobules, and this is the connective tissue septa which is dividing the part of this gland into different lobules. Now this is the one lobule which is having systemic this acinar part. Serous acini are present here. So within this, what type of gland is this? Which gland is this? What is the epithelium? It is serous, yes. And the duct system over here, as you can see, this is simple cuboidal. So simple cuboidal, and it is present within the lobule. So this is intralobular. Then here it is present between the lobules. This elongated duct which has simple columnar epithelium. This will be interlobular. So this is basically the exocrine part of the pancreas. You are going to see the different lobules divided by the connective septa. Each lobules have different serous acini. Then you must know how to explain serous acini. These are rounded pyramidal cells are there. They have rounded nucleus which is present towards the base. Apex has what? Why it is towards the base? Because apex is filled with zymogen granules. And then secretion इसका कहाँ जाएगा? First into the intercalated, then into the intralobular, then into the interlobular, and finally into the main pancreatic duct. And as the secretions are being poured into the duct system, so that is why this part of the gland is exocrine. And exocrine secretions are basically pancreatic juice having digestive enzyme and bicarbonate ions. Yes. Now what type of gland is this? Duct is this? It is running in between the connective tissue septa, interlobular. Epithelium here will be simple columnar epithelium. Now these are the two different parts. The outer one is exo, that is exocrine part that we have discussed. Now the second part of this is endocrine part. Endocrine part has clusters of cells or the cords of cells which are of basically three type, and we need special stain to identify these alpha, beta and delta cells. Alpha cells जो होते हैं islets of Langerhans में, they basically secrete which hormone? Glucagon hormone. And beta cells insulin, and delta cells somatostatin. Somatostatin क्या करेगा? It will inhibit the secretion of insulin and glucagon and growth hormone. Yes. So in between these serous acini, you are going to see these clusters of cells which are there. So these are the islets of Langerhans. Then what are the three types? Alpha, beta and gamma. What are the locations of alpha, beta and gamma? Alpha type islets of Langerhans are basically located at the periphery. these are stained red with the special stain, and these are present at the periphery. Then we have beta cells which are present in the center, which secretes insulin, and delta cells which are basically stained brown with the special stain. They are present also at the periphery. So delta cells and alpha cells are present at periphery, and beta cells are present in the center. What is the function of insulin? It will decrease the blood glucose level and glucagon. So endocrine part is basically islets, Langerhans. These have 3 cell types. Now can you recognize islets of Langerhans? So this is the second differentiating feature. In the parotid, you are not going to see these islets of Langerhans. Okay, and here in the this pancreas slide, there is no striated duct as there in the parotid. So these are the differentiating features. First one was centroacinar cell are there in the pancreas, and myoepithelial cells are there in the parotid. Here centroacinar cells, then islets of Langerhans are there, not there in the parotid, and the last one is striated duct is not here. There is intercalated, intralobular, interlobular, no striated duct here in the pancreas. These are the cell types. You can write down if you want to. 70% have beta cells in the center, delta cells are again present at the periphery, and alpha cells basically glucagon. 20% of the cell population of islets of Langerhans present at the periphery. तो बेसिकली अगर हिस्टोलॉजी ऑफ पेनक्रियास आपसे पूछा जाए या फोकस जब करने के लिए बोले तो आप कैसे आइडेंटिफाई करोगे उसको? When you put the slide under the microscope at 10x, you are going to see there are numerous serous acini, and they are separated by the connective tissue septa. There are lobules of glands which are there. Connective tissue septa have these elongated duct system also. This may be either pancreas or parotid. Then you focus more and you can see that there are clusters of cells in between these lobules. Okay, these can be islets of Langerhans. So this is the slide of pancreas. Yes. And when you focus on 40x in the lumen, you are going to see centroacinar cells which are not there in the parotid. So we have both exocrine, endocrine part explained here. Now the next slide we have of liver. This is very beautiful slide, one of my favorite topics, histology of liver. So liver is the largest or second largest organ? Second largest and second heaviest organ in the body. First largest skin, and internal largest organ is liver. It is wedge-shaped organ. It is present mostly in the right hypochondrium. It extends till the left hypochondrium. Mostly it is present in the right hypochondrium below right dome of the diaphragm. It has right lobe, left lobe, quadrate lobe, and caudate lobe. Next we have functions of liver. What are the functions of liver? Anyone? Please raise your hand. What are the functions of liver? Yes, detoxification or synthesis and secretion of bile juice. Then see, synthesis and secretion of bile. Then synthesis and secretion of plasma proteins like albumin, fibrinogen, and storage of glucose in the form of glycogen, excretion of bilirubin. Bilirubin कैसे excrete out होता है, कैसे form होता है, ये अभी आपका हुआ कवर टॉपिक UGCR में, जब आप लंच कर रहे थे सीट के नीचे. कहाँ गई वो? Yes, this girl, stand up. कैसे फॉर्म होता है बिलीरुबिन? Due to the worn out RBCs which are phagocytosed by the Kupffer cells. Sit down. Excretion of bilirubin, synthesis and secretion of bile, then synthesis and secretion of plasma proteins, then storage of glucose, detoxification of drugs and toxic products. These are some of the functions of liver. So basically liver जो है वो important function जो play कर रहा है वो metabolism का function है, metabolism of all carbohydrates, lipids and fats. Yes, and so it is an active organ. एक्टिव है ये, तो इसमें जो सेल ऑर्गेनेलीज होंगी, मोस्ट ऑफ द सेल ऑर्गेनेलीज आर देयर इन द साइटोप्लाज्म ऑफ हेपेटोसाइट्स. द स्ट्रक्चरल एंड फंक्शनल यूनिट ऑफ लीवर इज hepatocytes, which are cells of liver. These are polygonal in shape and they have abundant mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum and Golgi apparatus, because they are actively functioning. They are playing the role, important role in the body, and they are active, so they have abundant cell organelles. Now regeneration capacity of liver is high. That is why a small section, if it is being damaged, then it can be replaced by the proliferation of the hepatocytes, and that is why there is success rate in the transplantation of liver. And in the fetus, it plays an important role in the hemopoiesis. Histological organization of liver. Why I was telling this is one of my favorite, because it appears like this. No, not the colors are not there, but the arrangement is this. So basically it has a connective tissue capsule which is known as Gleason's capsule. Now this Gleason's capsule is thickened at the entrance of the structures which are present at porta hepatis, portal vein, hepatic artery and bile duct. Now this connective tissue again sends the connective tissue septa into the liver and divides the liver into incomplete lobules. Yes, and these lobules have radially arranged cords of hepatocytes. So liver is enclosed by a capsule which is basically dense fibroelastic capsule. It is known as Gleason's capsule. Then it sends the septa into the liver and divides it into number of incomplete lobules. Now the parenchyma part of liver, it is supported by type 3rd collagen fibers, that is reticulin, and it gives a structural framework to the hepatocytes, which are the structural and functional unit of liver. Now to start with, this is the classical liver hepatic lobule. Here there is a central vein in the center. This is the central vein. ठीक है? अब यहाँ से radially arranged हैं cords of hepatocytes. These are running from the center towards the periphery. Now there is a hexagonal arrangement of each of these classical hepatic lobule. Hexagonal means which have 6 sides, and at each angle there are present portal triad or the portal tract or portal canal. Portal canal have the branch of portal vein, branch of hepatic artery and bile duct. So basically we have portal venule, not portal vein, because portal vein is present at the entrance, and when it enters into the liver, start dividing. So we at the portal triad we have portal venule, then we have hepatic arteriole and bile ductule. These are present at the 6 corners of this hexagonal arrangement of hepatocyte. This is the classical hepatic lobule. So what we are discussing, what are these cells? These are hepatocytes. Hepatocytes are the cells which are there in the liver, in the parenchyma of the liver. Then these hepatocytes, if they are arranged around central vein, they are radiating out, present in the rows, and at each angle of this hexagonal part it has a portal triad or a portal tract, because these are the connective tissue septa, and in connective tissue septa these are covered, these structures are covered. These are portal venule, hepatic arteriole and bile ductule. Now this arrangement is basically a classical hepatic lobule. In between these hepatocytes, there are blood spaces, open blood spaces, which are known as sinusoids. These sinusoids are lined by discontinuous fenestrated endothelium. Endothelium is simple squamous. Fenestrated मतलब क्या होता है, इसमें pores होते हैं, and discontinuous मतलब it is not incomplete, कहीं कहीं there are gaps. Now this type of epithelium basically it is providing opportunity to the nutrient which are being absorbed from the GIT to get into the hepatocytes. Yes, basically यहाँ पे blood कहाँ से आ रहा है? From the portal vein. Portal vein is carrying blood from the intestine, spleen, pancreas. Yes, and basically it is these are containing the important nutrients which are being absorbed in the GIT. Yes, so 70% of the blood supply of liver is by the portal vein. Mostly it is supplied by the portal vein, and remaining 30% by the hepatic artery. Now both of these keep dividing and then they open into the sinusoids. Okay, now the blood here in the sinusoid, it runs towards the center, towards the central vein. जो हमने central vein बनाई है, इसकी तरफ यहाँ ये sinusoid में क्या open हुआ? ये जो artery और vein है, इनमें जो blood है, it opens into the sinusoid and runs towards the central vein. Now these central vein combine and they will form sublobular vein. Sublobular vein will combine and form hepatic veins, and hepatic veins will finally drain into. फोन बंद कर दे या क्लास से बाहर चली जाओ, या फेंक दूं फोन को मैं. Now the central vein drains into sublobular vein. Sublobular vein combines and form hepatic veins, and hepatic veins drains into inferior vena cava. So finally the blood from the liver is drained into the inferior vena cava. Okay. So this kind of arrangement of hepatocytes around central vein, having 6 corners, having 6 portal triads and sinusoids in between, which is lined by discontinuous fenestrated endothelium. This kind of arrangement is of classical hepatic lobule. Basically blood जो है, it is drained into sinusoids and then it is being passed into the central vein, and the important nutrients are absorbed, percolates into the hepatocyte through the sinusoids. Now the second arrangement, which is the functional arrangement, one is the portal lobule and another is hepatic acinus. Now this portal lobule is triangular. इसमें आपको तीनों classical lobule भी दिखा रखा है, इसके अंदर portal lobule भी दिखा रखा है और hepatic acinus भी दिखा रखा है. तो हमने जो अभी डिस्कस किया था वो क्या डिस्कस किया था? सेंट्रल, ये एक ये क्या है? दीज आर द क्लासिकल हेपाटिक लोब्यूल. These are the central part has central vein. Okay, then we have this triangular lobule which has this portal triad in the center and at each corner we have central vein. इसमें क्या है? ये जो एक पूरा यूनिट का जो बाइल बनेगा वो कहाँ जाएगा? into this bile ductule. Okay, तो ये यूनिट का बाइल किस में जा रहा है? bile ductule. So it is basically denoting the formation, secretion of secretion of hepatocytes into the bile ductule through all these lobules. Okay, now the last one is hepatic acinus. Again this is functional lobule. यहाँ पर ये क्या शो करने की कोशिश कर रहे हैं? यहाँ पे जो सेंटर में जो ब्लड है, now this blood is being passed on into these cells. So blood here is via hepatic artery and portal vein. Okay, they will release blood into the sinusoids and they are present here in the center and then they are being absorbed from the hepatocytes here. So this hepatic acinus is basically diamond shaped unit or a hepatic lobule which has a short axis which connects to portal triad and a long axis which connects to central vein. And the cells which are present here in the center, they are basically getting maximum amount of nutrients because they are present near to these blood which is being drained here. तो यहाँ पे जो न्यूट्रिएंट एब्जॉर्प्शन होगा मैक्सिमम होगा, ऑक्सीजन एब्जॉर्प्शन होगा और यहाँ पर जो साथ में टॉक्सिन्स आ रहे हैं वो भी सबसे ज्यादा कौन लेगा, ये जोन 1 वाले सेल। फिर इसके पास में सेंट्रल जोन वाले सेल होते हैं, देयर आर जोन 2 सेल्स, एंड सबसे बाहर वाले जो सेल्स होते हैं वो होते हैं जोन 3 सेल्स। एंड जोन 3 सेल्स में सबसे कम न्यूट्रिएंट पास ऑन होगा और सबसे कम टॉक्सिन पास होगा, ठीक है। और सबसे ज्यादा न्यूट्रिएंट टॉक्सिन कौन से सेल्स के पास होगा, जोन 1 सेल्स। So these are the structural and functional unit of liver arrangement of hepatocytes. That first is basically the blood flow from into the sinusoid. Second is the secretion of the bile juice into the bile duct, and third is the absorption of blood into the hepatocytes. So they are formation of 3 zones. Now what are these units? The first being the structural unit is the hexagonal arrangement, that is classical hepatic lobule. Second, we have this triangular arrangement, this is portal, and last one is hepatic. तो क्लासिक के अंदर क्या है, सेंटर में वी हैव सेंट्रल वेन एंड 6 कॉर्नर्स पे वी हैव पोर्टल ट्राइड। पोर्टल ट्राइड में क्या ओपन हो रहा है, देयर इज पोर्टल, यस, ब्रांच ऑफ पोर्टल वेन, पोर्टल, हेपेटिक आर्टरी एंड बाइल डक्ट। नो हेपेटिक वेन। हेपेटिक वेन कैसे बनेगी, सेंट्रल वेन से ब्लड कहाँ जाएगा, सब लोबुलर, सब लोबुलर से फिर हेपेटिक वेन्स में। तो पोर्टल ट्राइड में देयर इज नो हेपेटिक वेन, देयर इज पोर्टल वेन, हेपेटिक आर्टरी एंड बाइल डक्ट ट्यूब। Okay, then here in the triangular part, this is the portal lobule which has this central vein at the corners and portal triad here in the center, and we have hepatic acinus which has these short axis connecting the portal triad, long axis connecting the central vein, and basically it is denoting the absorption of the nutrients through the blood from the sinusoids by the hepatocytes. The first one is classical lobule. Are you clear with this? What is classical lobule and what is portal lobule? Any pathology is denoted here in the this portal lobule, which is basically it is having this in the center, it is having portal triad, and the last again it is pathologically important. Why? Because जो भी होगा यहाँ जोन में, अगर टॉक्सिन्स आ रहे हैं तो सबसे पहले कौन से सेल कॉम्प्रोमाइज होंगे, जोन 1 सेल्स. So these are basically more important functionally. This one, portal lobule and liver acinus. You can take the picture if you want to. Any problem in understanding this portal lobule, then liver acinus? These are the 3 zones donated here. Maximum nutrients being absorbed by the zone 1 cells, then zone 2, and then lastly zone 3. So ये तो हो गया arrangements of hepatocytes and arrangement of the lobules. Now what else? What we are going to see? Hepatocytes. These are present in the form of cords, radially arranged, and they have adjacent sinusoids. Well and good. Now what is space of disc? Space of disc is basically space which is there between the sinusoids and hepatocytes. Hepatocytes और sinusoids के बीच में जो space है, that is space of disc. MCQ पूछा जाता है, space of disc is present in liver, kidney, gallbladder, pancreas. Okay, so space of disc is basically space between sinusoids and hepatocytes. Space of mole is space which is present between the portal triad and hepatocytes. Hepatocytes और portal triad के बीच में जो space होगा, that is known as space of mole. Now bile canaliculi, ये हम डायग्राम से समझेंगे बाइल कैनालिक्यूली कैसे फॉर्म हो रही है, और अभी जो अपना टॉपिक था उसमें भी एक्सप्लेन किया गया था आपके सीनियर के थ्रू. देन पोर्टल ट्रैक्ट बेसिकली हैव व्हाट, पैसेज ऑफ पोर्टल वेन, ब्रांच ऑफ पोर्टल वेन, पोर्टल हेपेटिक आर्टरी एंड bile duct. Central vein and its drainage I already explained you. Now let's see in the figures. ये क्या जा रहा है? This is the movement of blood from the sinusoids towards the central vein. तो इस टाइप के फ्लो को या डायरेक्शन को क्या बोलते हैं? तो सेंटर की तरफ जाता है, Petal towards the center. This is centripetal. So direction of blood is basically towards the central vein. And what is this direction? This is going away from the center. So this is green in color, centrifugal. This is green in color. So it is denoting what? Yes, bile. So this is secretion of bile and it is basically away from the central vein. So its direction is centrifugal. ठीक है, vein sinusoids में जो blood है, it is going towards the central vein and this bile juice is basically secreted out in a direction opposite to that of central vein. So direction is centrifugal for the bile juice and centripetal for the sinusoids, blood in the sinusoids. Now this is how this classical lobule appears here under the microscope. ठीक है, जैसे ही आप साइड फोकस करेंगे, you are going to see the central, numerous central vein are there. At the corners there is this dilated part. Now please tell me, you have read the histology of blood vessels. Now there is this dilated lumen having simple square. Yes, this is portal venule. Very good. And then we have this limited lumen arteriole which is hepatic, and then ductule, जहाँ पे भी ducts आते हैं, ductules आते हैं, their epithelium either low cuboidal or columnar. So this is basically what? Bile duct or bile ductule. ठीक है, किसी को प्रॉब्लम ये समझने में? This is large lumen of portal vein, then we have small limited lumen of hepatic artery, and then we have simple cuboidal epithelium for bile duct. So this represents what? Portal triad. So what will be the epithelium of portal vein? Endothelium. जहाँ पे भी word endothelium, mesothelium आता है, they are all lined by simple squamous epithelium. So for the artery and vein it is simple squamous epithelium. For duct it is simple cuboidal. Now please tell me, what is this in the center? Now what is this arrangement of cells? Yes, radially arranged hepatocytes. Now this what does this denote? Sinusoids. Now the space between the sinusoids and this will be space of this, and space here between the hepatocytes and this portal triad will be space of Morse. Now in the liver of pig, this is well demarcated and easily differentiated, each of the classical lobule, but this is not easily differentiated here in the human liver because incomplete division of the septa. So first you have to locate these portal triad and join them with the imaginary line to discover one hexagonal structure, that is classical liver lobule. आप क्या करेंगे? Central vein locate करेंगे and then you are going to locate portal triad. जहाँ पे यह 6 portal triad को connect करेंगे तो एक classical lobule बन जाएगा. This is polygonal hepatocytes. This has this rounded nucleus, then it has 1 to 2 nuclei actively they are participating in the metabolism and detoxification. Bile canaliculus. Now surfaces of these hepatocytes, like 2 surfaces are enclosing this open space here. This is sinusoid. Now this space is space of this. Now this is the surface of hepatocytes. There are microvilli projecting towards this space, because what is the function of this surface? It has to absorb the nutrients and oxygen from the bloodstream over here. Okay, so this is going to be percolated through this and come into in the space of this and then into the surface of the hepatocytes. So this is basically arrangement of what does it show? This blue color, ये क्या है? This is the epithelium lining of the sinusoid, which is basically fenestrated this continuous endothelium, and then this is space of this, and these are the microvilli present on this. So these 2 surfaces are enclosing this sinusoids. Now there are 2 adjacent hepatocytes. They have grooves on their both parts in the plasma membrane of adjacent hepatocytes, and at the corners these are tightened by the gap junctions. This particular part is basically your starting point of duct system for the secretion of bile. Bile canaliculi. Bile canaliculi का अलग से कोई border नहीं है. This border is formed by the plasma membrane of two adjacent hepatocytes. जो भी formation होगा that will first secreted out here into the bile canaliculus. Then from the bile canaliculus through the canal of Herring it will go into the hepatic ductus. They will combine and form large hepatic ducts, and hepatic ducts will form common, right and left will form common hepatic ducts. Then common hepatic ducts are going to combine with the, how common bile duct is going to form? Yes, so with the duct of gallbladder, which is cystic duct, and it's going to form common bile duct. Common bile duct then it's going to open along with the pancreas, main pancreatic duct, into the second part of duodenum at the ampulla of Vater. And rest these junctions are closed by the junctional complex. So these are the basically surfaces of the neighboring hepatocyte. Two of them are enclosing the sinusoids, two adjacent are having the bile canaliculi, and rest surfaces are being enclosed. So basically यहाँ से कोई लीकेज नहीं होगा क्योंकि यहाँ पे क्या है, these are enclosed by this junctional complex. यहाँ पर भी ये tightened है, only here the secretion is going to pass down into the bile canaliculus. This is the sinusoidal, I already explained. Now, in addition to the endothelium cells, here in the sinusoids, we have one more modification of cell, phagocytic cell, which is there in the liver, which is Kupffer cell. So, this is the discontinuation lining, which is endothelium lining. In between this discontinuous lining, we have enlarged macrophages, which are known as Kupffer cell. Basically, Kupffer cell का function क्या है? Phagocytosis of the cellular debris, clearing out the bloodstream. So here Kupffer cell is basically representing monocytic phagocytic system. It is macrophage present in the liver, in the gaps between this epithelium of the endothelium. Then next we have space of disc, already explained. Lastly we have etocells. These are present in the space of disc and they basically store lipids and vitamin A. And also when liver is damaged, they multiply and form fibroblast, kind of fibroblast. So basically the nodular or the fibrotic mass of the liver is formed if they are highly activated, if liver is damaged. Their main function is the storage of vitamin A and lipids. And etocells are there, these are the stellate cells which are there in the space of disc. ठीक है, space of disc कहाँ है? Adjacent to the hepatocyte and sinusoid. See, through this bile canaliculi, the blood will flow into this canal, which is canal of Herring, and then into the ductule. And the flow will be opposite that of the venous blood in the sinusoids, opposite to the center. Again, this diagram represents the opposite flow of the blood. This is the drainage of the blood from the central vein into the sublobular vein and then into hepatic and then inferior vena cava. इतना समझ में आया सबको? So we have completed histology of liver. It is mostly it is likely to come in the university exam. Histology of liver is the most common question, and it is one of my favorites. I don't know if you are able to understand or not, but this is really beautiful slide of liver. Have you understood it? I tried my best. This is up to you. Now next we have gallbladder. Now what is the function of gallbladder? Where it is located? See, basically it is present in the association with liver. So it is present on the visceral surface. There is a fossa for the gallbladder on the surface of the liver where it is present, and it stores bile and concentrates the bile, and it secretes as and when it is required. What is the function of bile juice? emulsification of fat. So when it is gets stimulated, it's going to secrete the bile that is going to be secreted in the second part of duodenum, जहाँ से क्या आ रहा है, food is coming from what part? Stomach, which is highly acidic. So secretion from here are alkaline in nature. Secretion from the pancreas is all again alkaline in nature. So this is going to neutralize the acidic chyme which is coming from the stomach. Do you remember the layers that we have studied in the GIT? There is innermost is mucosa. Mucosa has this lamina propria and muscularis mucosa. Then we have submucosa, muscularis externa and either serosa or adventitia, depending upon if it is covered by peritoneum or not. Yes, here in gallbladder we don't have this muscularis mucosa and submucosa. Okay, there is no muscularis mucosa and submucosa. See, the inner part which is holding this concentrating the bile juice, it is lined by mucosa which is basically simple columnar epithelium. GIT का क्या था? Epithelium simple columnar, and it has a brush border. Brush border मतलब it has microvilli on its surface, because यहाँ पे क्या काम है इसका? Absorption of water, concentration of bile. So it has a brush border simple columnar epithelium. Then underlying mucosa have lamina, which has lamina propria, which has number of elastic fibers, and then we have a fibromuscular coat. There is no muscularis this mucosa. There is this fibromuscular coat which has disorganized smooth muscle cells, and there are collagen fibers and elastic fibers in that fibromuscular coat. There is no muscularis mucosa or submucosa. And lastly we have either adventitia or serosa. So adventitia कहाँ होगा? When it is covered by peritoneum. So basically the lower part, the inferior surface which is in contact with the peritoneum, and the fundic part of gallbladder is covered with serosa, and the part which is covered by the liver, which is where it is situated, the upper part which is in the fossa for the gallbladder, it is covered by adventitia. So depending upon its surface, if it is upper surface, it is covered by adventitia. If it is lower surface, it is covered by serosa. Serosa and adventitia are the outermost connective tissue layers which have these blood vessels and lymphatic. Serosa also have peritoneum simple squamous epithelium, yes, which is derived because peritoneum is basically derived from the mesothelium. So let's see the histology of this gallbladder. We have folds of mucosa. These are the temporary fold of mucosa when this gallbladder is empty. When it is distended, these folds disappear. And these folds have this simple columnar brush border epithelium. Where else you are going to see this brush border epithelium? Yes, brush border epithelium. और कहाँ देखी आपने? Microvilli कहाँ देखे हैं आपने? इलियम में, small intestine के अंदर. So here also it is brush border epithelium. Then you have this connective tissue layer which is lamina propria. Now this is muscularis fibromuscular coat, and there are interspaced collagen fibers there in between, and elastic fibers are there. That is why the name fibromuscular coat. कौन सा layer नहीं होगा? Submucosa नहीं होगा and muscularis mucosa. Outermost this connective tissue layer, depending upon the surface, is either adventitia or serosa. What are the features of simple columnar epithelium? Elongated cells, nucleus towards the base, yes, and here on the upper part what they are going to show? Microvilli. And because of this, this epithelium is known as brush bordered epithelium. Now differentiating feature from the ileum or the small intestine, there are no goblet cells, there is no submucosa, there is no muscularis mucosa. And in the intestine we have seen that there are arrangement of circular and longitudinal layers. There is no such arrangement, no organized arrangement of smooth muscle cells, disorganized arrangement of these smooth muscle cells. So these are the differentiating feature of gallbladder from the small intestine. Small intestine, large intestine have number of goblet cells basically. We are done with this. So what topic we have covered today? Histology of pancreas, liver and gallbladder. Pancreas and gallbladder तो छोटे ही है, liver की histology थोड़ी complicated है. ये किसका स्लाइड है? कैसे आइडेंटिफाई करोगे आपकी लीवर है? ऊपर लिखा है, ऐसे स्लाइड पे भी लिखा होगा, एग्जामिनर पूछेगा हाउ यू आर गोइंग टू आइडेंटिफाई, हाउ यू हैव आइडेंटिफाइड इट, टेल मी विच स्लाइड इज दिस, यू आर गोइंग टू से लीवर एंड देन radial arrangement of what? Hepatocytes, which are its structural and functional units. So they are radiating out from what? So what is these areas, these circular, these are representing central vein, and these are radiating out to outwards in the form of sheets. These are sheets of hepatocytes, and then adjacent part they have again this slightly stained areas. These are sinusoids. Sinusoids are lined by endothelium, discontinuous fenestrated endothelium, and the sinusoids are basically what? Blood filled spaces. Now see, portal vein, this portal vein, hepatic artery and bile duct, they enter into the liver from its visceral surface, and then they keep on dividing. Bile duct will come out, and these structures they keep on dividing until unless they open into the sinusoids. So this blood from the portal vein and this hepatic artery then drains into the sinusoids. Important contents are absorbed by the hepatocytes through space of disc. Space of disc is present between hepatocytes and sinusoids. Now, in between the lining of the sinusoids, there are modified cells, macrophages are there, which are known as Kupffer cells. What is the function of Kupffer cell? Phagocytosis of cellular debris. In the space of this space of this, there is again there is this rounded structures, cells there, which are there, which are known as, not rounded, these are basically stellate shaped. These are itocells. Itocells function is to store vitamin A and lipids. The hepatocytes will enclose sinusoids. Adjacent hepatocytes also have bile canaliculi, where the secretion of bile is being done. Then it is transferred through a canal which is known as canal of Herring into the hepatic ductules. Hepatic ductules will combine and form right and left hepatic ducts. So right will come from the right part and the left from the left part, and then they will form common hepatic duct. Common hepatic duct comes out at porta hepatis, the point where these structures are present: portal vein, hepatic artery and bile, this common hepatic duct. Now here at the 6 corners, what you are going to see? Portal triad. What are the structure present in the portal triad? Portal vein पूरा तो होगा नहीं, this branch, so you can say portal venule, hepatic arteriole and bile ductule. Epithelium of portal vein and hepatic arteries and bile ductule locuboidal. Then ये तो हमारा क्या है? क्लासिकल लोब्यूल. उसके बाद हमने एक पोर्टल लोब्यूल पढ़ा, वो समझ में आया? वो बेसिकली एक ट्राइंगुलर, अगर हम ये 3 एरियाज को ज्वाइन कर दें सेंट्रल वेन को, यहाँ पे एक जो ये पोर्टल ट्राइड इसको सेंटर में प्रेजेंट होगा, तो ये क्या बनाएगा? पोर्टल लोब्यूल. एंड व्हाट इज हेपैटिक एसिनस? अगर हम ये 2 सेंट्रल वेन को जोड़ दें तो और 2 पोर्टल लोब्यूल को जोड़ दें तो ये क्या बन जाएगा अपना? हेपैटिक एसिनस. एक यूनिट बन जाएगा. और ये यूनिट क्या रिप्रेजेंट करेगा? इनके बीच में क्या होगा? सेंट्रली प्रेजेंट ब्लड which is being absorbed through the hepatocytes. So mostly it is absorbed by the zone 1 cells and least absorbed by the zone 3 cells of hepatic acid. Anything else that we have read today in the section of liver that is left to discuss? Anything else? Classical, so we have already discussed. This is the classical part. सबसे पहले आप फोकस करते ही क्या करोगे? रिप्रेजेंट की दिस इज द क्लासिकल हेपैटिक लोब्यूल. जहाँ से ये रेडिएटिंग आपने बोला इट्स कमिंग आउट, अरेंजमेंट्स ऑफ हेपैटोसाइट्स, बेसिकली दिस इज क्लासिकल हेपैटिक लोब्यूल. So you must know what are the content of portal triad. These are present at the angles, 6 angles of this hexagonal structures. Central vein, what is सेंट्रल वेन का क्या फंक्शन है? बेसिकली जो यहाँ पे जो एब्जॉर्ब होना है, इंपॉर्टेंट कंटेंट्स बीइंग एब्जॉर्ब, देन वेस्ट विल गो इनटू द सेंट्रल वेन. थ्रू सेंट्रल वेन दिस ब्लड इज गोइंग टू ड्रेन इनटू सबलोबुलर, थ्रू इनटू द हेपैटिक एंड फाइनली इनटू द इंफीरियर वेना केवा. ये क्या है? classical hepatic lobule. Now which slide is this? Pancreas. ये क्या है pancreas में? These are exocrine part represented by, ये न्यूक्लियस है ना, और ये इधर ये सेंट्रोएसिना, तो these are serous acini. एंड ये जो पार्ट है, और यहाँ पे कितने टाइप्स के हैं? बेसिकली 3 टाइप्स. और भी है, अगर स्पेशल स्टेन यूज करें तो पीपी टाइप्स और भी सेल्स मिल जाएंगे. बेसिकली 3 टाइप्स फ्रॉम द सेंटर टू द आउटर पार्ट. इन द सेंटर वी हैव बीटा एंड देन वी हैव अल्फा एंड डेल्टा. बीटा सेल्स क्या सिक्रीट करेंगे? अल्फा. एंड लास्ट डेल्टा सेल्स. अगर कम सिक्रीशन हो रहा है इंसुलिन का तो क्या होगा? ग्लूकागोन का तो वो तो होगा ही, क्योंकि ग्लूकोज अगर कम है तो किसका सिक्रीशन ज्यादा होगा? ग्लूकागोन. और अगर ग्लूकोज ज्यादा है तो किसका सिक्रीशन बढ़ जाएगा? ये किसका स्लाइड है? कैसे पहचानोगे इसको? रेगुलर म्यूकोसा, सारे जीआईटी स्लाइड में रेगुलर म्यूकोसा दिख रहा है तुम्हें. इसमें सिंपल कॉलोनर सेल्स भी है और ब्रश बॉर्डर एपिथेलियम है, पर इसमें क्या नहीं है? इस तो मस्कुलरिस दिख जाएगा. कौन सा मस्कुलर? ये भी मसल जैसा ही दिख रहा है. ये सिंपल कॉलमनर सेल्स एपिथेलियम होंगे. यहाँ पे फोल्ड्स हैं, पर यहाँ पे गॉबलेट सेल्स नहीं है. गॉबलेट सेल्स कैसे दिखते हैं? इन बीच में आपको बिल्कुल स्टेनिंग नहीं दिखेगा, गैप्स दिखेंगे, है ना? वाई? बिकॉज म्यूकस तो वॉश आउट हो जाता है, स्टेन नहीं होता है. सो दैट इज वाई lightly stained cells are present in between, but this is continuous presence of epithelium is there. Columnar cells are there, brush border epithelium, no goblet cells. This may be slide of gallbladder. Let's look downwards now. This one is actually, there is no arrangement of uniform arrangement of muscular core. These are interspersed with the collagen fibers are there. So this is the slide of gallbladder. Now what is the capacity of gallbladder? What is the capacity of gallbladder? क्या का फंक्शन है गॉल ब्लैडर का? स्टोरेज ऑफ बाइल. तो कितना बाइल स्टोर कर सकता है? ये वाइवा में पूछा जाएगा आपसे. 50 to 100 ml. 100 ml 100 ml से ज्यादा स्टोर नहीं कर सकता. तो इसकी कैपेसिटी मैक्सिमम इज 100